Okay, this is the pre-lab lecture video for experiment 6, which is going to deal with chromatography. In this experiment, what we're going to do is we're going to use thin layer chromatography and have an introduction to column chromatography. And we're going to do these two techniques after we isolate some pigments from spinach leaves. And the pigments that we're going to get consist of both polar and non-polar components. So really what we're going to do is we're going to examine the role of polarity in thin layer chromatography, and then we're going to separate the pigments using column chromatography. And so I'm going to talk about the experimental procedure, and there's a second video that deals with sort of the theory of thin layer and column chromatography, just the basics. So we're going to be isolating different spinach pigments from raw spinach leaves. And so there's different classes of these. The first is a yellow pigment that we get that is completely non-polar. It's totally a hydrocarbon, and that is called beta-carotene. Beta-carotene is also in carrots. It's really the only pigment that you find in carrots. Then there's some other xanthophylls that have this carotene structure, only they have now polar groups, like hydroxyl groups, attached. And so these make up what are called xanthophylls, things like lutein and cryptoxanthin and zeaxanthin. But these would be a little bit more polar than the beta-carotene would be. And then the major classifications are pigments called chlorophylls. And if you've taken biology, you are probably familiar with the chlorophylls. They're the green pigments that are in leaves, and they're the green pigments in this case that are in the spinach leaves. The structure of the chlorophyll is it has this porphyrin group, which has 4 nitrogens, and those 4 nitrogens basically are coordinating to a magnesium 2+ ions. And then they have different functional groups attached depending on whether it's chlorophyll A or chlorophyll B. R1 is a CH3 and R2 is an ethyl group in chlorophyll A. And then a very similar structure has an aldehyde at R1. R3 is a long-chained group called the phytyl group, and so that makes the spinach have both a polar portion and a non-polar portion. And so the pigment, the way we're going to look at this is chlorophyll is going to be more polar than the carotene. Now, if you take away that magnesium from the porphyrin group, you end up with something called a pheophytin. And so this is the structure of pheophytin. It kind of shows that phytyl group a little bit clearer. There's a pheophytin A and a pheophytin B, depending on whether you started with chlorophyll A or chlorophyll B. But these groups are not as polar as the chlorophylls because now they've lost the ionic magnesium from their porphyrin group. It turns out this is actually this turns out to be important in food chemistry because if you have a green leafy vegetable and you put it inside of a can, then you want that green leafy vegetable to maintain as much of the green color with spinach or with this magnesium as possible. And sometimes under acidic conditions you can lose the magnesium, which means that your actual pigments would then turn from a green color to a grayish color. As we'll see in the experiment, the pheophytins are gray. And so you don't want to open up your can of spinach, which you probably don't want to do anyway, but if you did, you wouldn't want to see it gray. And so they have to somehow keep the magnesium in the porphyrin group in order to keep the vegetables nice and green. In the pre-1900s, they actually used to add copper to it to make the green it was sort of a weird green color. But then they found that adding copper to food was probably not a good idea. And in England in 1900, it kind of got banned from practice for health reasons. So these are the pigments that we're going to use. The non-polar beta-carotene, the somewhat more polar but still pretty much non-polar xanthophylls, and then we have our chlorophyll and our pheophytin without the magnesium. So when it's all said and done, we're going to be able to tell the difference between all of these pigments. We're going to see a beta carotene, we're going to see a pheophytin, we're going to see the two chlorophylls, chlorophyll A and chlorophyll B, and then some of the other pigments that we won't identify that are down here. Okay. So first we have to extract the spinach pigments. You're going to be doing this experiment in groups of 2 or 3 because it's easier that way and it'll save on resources. So the first step is we have to extract the pigments from the spinach leaves, and I have photos to show you exactly what you're going to do. But the first thing you're going to do is you're going to get fresh spinach leaves, and you're going to go ahead and you're going to tear off the stalk, and then you're going to place them in a mortar and pestle and crush them after adding some acetone. That acetone solution, then you're going to try and transfer the liquid without the solid mess into a centrifuge tube. We're going to add water and hexane in the proportions that the procedure suggests. You're going to get 2 layers. The top layer will be the hexane layer, the acetone and water will be the lower layer, and the pigments will be extracted into the hexane layer, which should become dark green in color. And then if an emulsion forms, which it will, because it's impossible to transfer the solution without getting some spinach stuff in there, then we're going to go ahead and centrifuge the centrifuge tube in order to break the emulsion. You're going to remove the upper green layer using a glass pipette. You're going to use glass pipettes in all of this experiment. You're going to place that into a conical vial, and then we're going to go ahead and dry it by preparing some drying tubes with small glass pipettes. We're going to pass 1/3 of the mixture through sodium sulfate, the other third of the mixture through magnesium sulfate, and you're going to keep 1/3 and not keep it in case you need to have more. And you'll see that the choice of drying reagent here is important because something will happen when you use magnesium sulfate that won't happen with sodium. You're then going to take the dried portion with the sodium sulfate, and we're going to determine the best TLC solvent for analysis. So here's what you're going to do. You're going to take fresh spinach, tear off the leaves, leave the stalks behind, add a milliliter or so of acetone, and then crush it with a mortar and pestle, and you'll see you'll get a nice green solution. You're going to transfer that green solution into a 15 milliliter centrifuge tube. You're going to add water and hexanes to this, which will give you 2 layers. And you just put the cap on the centrifuge tube and gently kind of rock it back and forth to do your extraction. We're then going to break the emulsion and get a clean line between the 2 layers by putting it in a centrifuge. And when we put these solutions in the centrifuge, we have to make sure that they are counterbalanced. So we have to make sure that there are 2 centrifuge tubes opposite each other as they spin, or else the rotor will wobble and then bad things happen. After it's been centrifuged, you'll clearly see the 2 layers. You want this top layer to be as dark green as possible. And during the experiment, we'll kind of be checking and making sure that that layer is nice and dark and green, because the more pigments, the more concentrated the solution, the better your TLC results are. This isn't glowing. It was next to the window, so you're just seeing the light come in from the window. It's not like it glows in the dark. You're going to take and use a glass pipette and remove the top layer then and place it into a 5 ml conical vial, so you can go ahead and put the cap on it to keep the solutions from evaporating. Now, to dry the sample, we're only going to have a milliliter or 2 of solution, and so it's really difficult sometimes to dry that thoroughly. So what we're going to do is prepare what we call a drying column. So you're going to have to take a glass pipette, and using your small clamp, you're going to clamp the glass pipette in place. Apparently, in the last couple of years, we've broken a number of the small clamps that actually fit and held the glass pipette snug. So you may have to get a small piece of rubber tubing that will have and then sort of slide that around in order to make this big enough to clamp in place. Didn't realize that until I just did this experiment. Then you're going to take a small piece of cotton and you're going to go ahead and make a little ball and push that down to the bottom of the glass pipette. If cotton balls freak you out, which apparently they do some people, then have somebody else do it. But you don't want to push this in too tight. You just want the cotton ball to kind of hold the sodium sulfate drying reagent and keep it from running out the bottom of the pipette. You're then going to add enough sodium sulfate to give you 1 inch to 1.5 inches of height, and you can make a little pipette out of or you can make a little funnel out of the plastic pipettes to help you pour the solution in. And then you're going to go ahead and add your 1/3 of your green extract to the drying tube. As you add it, the solution will drain down and you'll collect it in a small disposable test tube. To get the last little bit of liquid out of this, put a rubber bulb on top of the glass pipette and squeeze it, and you can push the rest of the solution out into the test tube. So now we have, and I'm using sodium sulfate in this part of the demonstration, we have sodium sulfate dried spinach extract. So then what we're going to do is we're going to determine the best TLC solvent to use. Now, when you get a TLC plate, you want to draw a line in pencil about 1 centimeter from the bottom, and then you put an X wherever you're going to place a spot. So you're going to create like lanes. This is a small test tube because I'm only making one. This is a small piece of TLC plate. You'll have bigger ones, so you'll have 3 or 4 X's across here. But this is a small one that I used for demonstration purposes. You have to do this in pencil because if you do this in pen, the pen ink will chromatograph, or it will be separated. It'll TLC exactly like the spinach pigments. So you don't want to do this in pen. You have to do it in pencil. Okay, because you don't want ink spots mixed in with your spinach spots. Then you're going to take a small capillary, which is really thin, and you're going to dip that into the solution. Some solution will then be drawn up, and when you touch the tip of that capillary to the TLC plate, the solution will then kind of transfer out. And you have to do this 5, 6, 7 times because you want that spot to be really dark, because the darker the spot, the better you'll be able to see the other spots as it separates during the TLC development. So you want to spot it probably 5, 6, up to 10 times to make sure that your spot is really, really as dark as you can get it. Okay. To develop the TLC plate, we take our TLC developing jar, which are in our hoods, and we always have to clean that jar out with acetone and make sure that the jar is thoroughly dry. This experiment is extremely sensitive to water, and so you want to make sure that you clean the TLC jar out and that you make sure it's dry, because otherwise you're going to contaminate your TLC plate, and I have no idea what kind of results you will get. You add probably 5, 6, 7 milliliters of solution. Actually, in this picture, I didn't add enough. You want the solution to cover the little bulge here in the bottom of the jar. So you want the solution to be smooth across. Then you're going to place your TLC plate in the jar. The reason that we use a 1 centimeter mark is simply because we don't want the spot immersed in the solution. So when you place the TLC plate in the jar, you're going to go ahead and cap it, and then the solvent will move up the TLC plate, separating the pigments as it moves. So here's sort of the beginning point. You can see that one band has separated, and there are some other bands down here. And as soon as the solvent has moved, let's say about 3/4 of the way up the plate, we'll consider that to be developed. And here you can see there's nice bands that we'll look at in further detail here. So as soon as the solvents move 3/4 of the way up the plate, you immediately pull the TLC plate out and you mark how far the solvent moved in pencil again, because that's how we're going to calculate our RF values, and we need to know how far the solvent moved. Obviously, we can measure how far the spot moved. So what we're going to do is, because these pigment spots will fade over time, is we're going to go ahead and take digital photographs of your TLC plates. Now, if you have a digital camera on your phone, feel free to take your own picture, because then you can email it to yourself and you can use it to make the measurements, or you can use it to make the measurements and print it out. But if not, we'll be taking digital pictures, and since each group is going to be doing this, we'll need to actually take pictures of this so everybody has a copy of the TLC plate and what it looked like. But nowadays, with everybody with their iPhones or their whatever phones, they have probably a better camera than I may use to take these pictures with. So you can see here that after the development, and this solvent, I got pretty close to being the best TLC solvent we could have. You can see that there's a band, there are different bands. It turns out that this band is the beta carotene. This gray band right here is the pheophytin. There's chlorophyll A and B, and then some other pigments that I personally haven't identified. And I know that these two bands are chlorophyll A and chlorophyll B because I know the color of chlorophyll A. It's this sort of blue-green color. For those of you in biology, this is chlorophyll A is the pigment that would be in your blue-green algae. And one of the ways that you can measure how much algae is in, let's say, a pond, is by collecting the water, sort of filtering off the algae, and then sort of crushing them, or actually extracting their pigments off into acetone, and then analyzing the green color that you get. I've done a little water chemistry, still do a little water chemistry, and that's typically how we've measured how much algae is in a lake. It's by measuring how much chlorophyll A we have. And in order to calibrate the instrument I use, I needed to order pure chlorophyll A, and so I know what its distinctive color is. Now, it turns out the chlorophyll A and B are fluorescent, so that if you took that TLC plate and you placed it under the black light that we used for our triboluminescent crystals, you could actually see that the chlorophyll A and B bands, as well as the xanthophyll band, glow red. And so they actually absorb ultraviolet light, and then they emit red light. You may say, why doesn't the chlorophyll A band glow as much? It's because there's something called fluorescence quenching, and so it's kind of quenching the fluorescence. And you can actually tell the difference between chlorophyll A and chlorophyll B if you use a fluorimeter, and I won't get into that, but it's actually pretty neat. You can tell how much of each of those there are. Even the xanthophyll is showing a little bit of fluorescence. So we won't do this, but just if you're curious, you can try it on your developed plates. Okay, so what we're going to do then to determine the best TLC solvent to use, and this has to be done before an analysis, and it pretty much is trial and error, is that you're going to basically prepare different hexanes, acetones mixtures. And then you're just going to simply vary the percentage of those, run TLC plates or develop TLC plates in each of those solvents, and see which one gives the best separation of the spinach pigments. Now, the one I used was pretty good. There may be a better one. And typically we make our chromatography solvents using a mixture of two solvents, and usually one is polar and one is non-polar. So that's why we're using hexanes and acetone in this case. So you're going to prepare 5 different mixtures: 1090 hexane, 3070, 30% hexane, 70% acetone, and then you're going to basically flip the percentages. So you're going to have 5 different solutions that contain different percentages of hexane and acetone. Real easy to make these solutions. You take a 10 milliliter graduated cylinder, you add 1 mil of hexane, 9 mils of acetone. Voila, 1090 mixture. Okay. So you're going to spot TLC plates, individual small ones, with the spinach pigment mixture, and then you're going to develop each plate in each one of the solvent mixtures. And the best solvent's the one that's going to separate the components the best. So you may get results that look like this. I don't have the plates labeled as to which solvent it is, but you can see that some of these solvents are better than others. And it's not just we're going to determine the best solvent, we're going to have to explain what happened in the solvents that didn't work the best. Why is this yellow pigment up here, and why is the green pigment here, and why do we see the transition that we see? Okay, and so that's what we're really going to investigate here in this experiment. And as I said in the last, or if you haven't watched, when you do watch the TLC sort of the chromatography tutorial, in biology, when you do these kinds of experiments, you're doing gel electrophoresis. Gel electrophoresis separates on the basis of molecular weight. In organic chemistry, we are separating on the basis of polarity. And so the polarity of the component, the polarity of the solvent, and the polarity of the plate make a really huge difference in terms of where the spots show up and how good your separation is. So you want to keep that in mind. The separations are based on polarity, not molecular weight. That's gel electrophoresis from biology. Okay. So then after you determine what the best TLC solvent is, you're going to see what different spot size makes. So you're going to have approximately a 2 millimeter, 5 millimeter, and 10 millimeter spot, and you'll see what should the optimum spot size be. If the spot's too big, do all the spots run together? And so you're going to investigate the spot size. And then we're going to do a very quick column chromatography experiment. In a few weeks, you will do a much more tedious one as we separate the components of an Excedrin tablet. But in this case, we're just going to prepare a small column, and the goal is to separate the beta-carotene from the rest of the chlorophylls. So you're going to take a glass pipette, and you're going to prepare a column exactly like you did with the drying column, except you're going to use alumina in this case, and alumina is aluminum oxide. Then you're going to fill the column with hexane, and then the hexane will then flow through the column to wet it. And then once there's just a little bit of hexane left at the top of the column, you're going to go ahead and add the pigment extract, and beta-carotene will actually move down the column and elute while the chlorophylls are stuck to the column. And as the beta-carotene elutes, you're going to collect that into its own test tube. Then you're going to add pure acetone to the column, and that will bring the chlorophylls out. That will elute them off the column into a test tube so that we'll be able to separate the beta-carotene from the chlorophylls as part of this experiment. So you prepare your column the same way you did with the sodium sulfate. I got a little plug of cotton here. I've got about 1 inch to 2 inches of alumina. I'm going to go ahead and add hexane to that and allow the hexane to wet and then start dripping off the column. As soon as there's only a little bit of hexane left at the top of the column, I'm going to go ahead and pipette in my sodium sulfate dried extract. And then I'm going to go ahead and it will basically flow down the column. And as it does, you'll see that the beta-carotene pigment begins to move down the column, whereas all the chlorophylls are stuck up here. Once the extract solution gets close to the top, you're going to go ahead and add more pure hexanes to this column, probably just a few milliliters at a time. Eventually this beta carotene band will move down the column and it'll start to elute off of it. Now you're going to collect the clear solution that's in this, that's coming out of this pipette. You're going to collect that clear solution until the band, just the yellow band, just starts to get close to the end. Then you're going to switch to a brand new test tube and you're going to collect the yellow band as it comes off the column. With pure hexanes, only the yellow band will come off. The green band may spread out, but it won't make it all the way down to the bottom. So as soon as the yellow band has been completely collected, you're going to go ahead and put a new test tube on there, or you can put the test tube that collected the clear solution at the beginning back on. Then you're going to go ahead and when the solution gets down, when the hexanes gets close to the top of the column, you're going to go ahead and add pure acetone. And as you add pure acetone and it moves down the column, it will take the chlorophylls and it will begin to elute those down the column. And as it elutes those down the column, when the green band just gets to the point where it's going to come off the column, you're going to switch, put a new test tube on there, and collect the green band. And at the end, after all the acetone has flown through here, and you may need to add some more acetone, but you're going to elute the green band completely off the column. And so in the end, you're going to have the carotenes in one test tube and the chlorophylls in the other. You're then going to TLC on one plate the spots of the original sodium sulfate dried solution, the solution after it went through the magnesium sulfate, and then you're going to analyze each one of those two fractions to show that indeed you've got beta carotene in one and chlorophyll in the other. Be interesting to see where the xanthophylls show up in all of this, or maybe they showed up, maybe they actually were collected in one of the clear layers that we did, or one of the clear fractions that we actually aren't going to analyze. Okay, so that's what we're going to do in this experiment. Prepare the extract and then do TLC and this column chromatography to then separate the pigments out. Okay. At the end of the experiment, you're going to go ahead and put all of your solutions in the waste container. You can go ahead and put the test tubes, the disposable test tubes, and the pipettes all in the glass waste container. We will take digital pictures, or you will take digital pictures of the plates so that you have pictures of those to be able to answer questions about them in your report form. And outside of that, that's our introduction to TLC and column chromatography. We'll use TLC quite a bit to determine how many components we have and sometimes identify the components in our TLC chromatogram, or in our TLC plates. And then we will do a large column extraction of the Excedrin tablet components in a few weeks, where we're going to collect 30 or 40 fractions, and in that case the components won't have a color to them, so we'll have to figure out exactly where those components are by using TLC. So that'll be a much more in-depth use of the column chromatography.